Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Kia ora koutou katoa. Welcome to Scigest. I'm your host, Hilary Ireland. Today on the Science Life for Me series, we have a very special guest. Principal Scientist at Plant and Food Research and Professor in the School of Biological Sciences at the University of Auckland, Professor Andrew Allan. Professor Allan was recently elected as a Fellow to the Academy of the Royal Society Te Apārangi in recognition of more than 30 years' research in plant genetics, and has featured on the Clarivate Highly-Cited Researchers list for seven years running, from 2017 to 2023. This is a list compiled from the world's population of scientists. It is therefore a huge privilege and pleasure to welcome to the podcast Professor Andrew Allan. Andy, welcome to Scigest.
Thank you very much. It's always a pleasure to chat.
Great to have you here. When you read or hear these accolades, what do you think?
Well, I know it sounds a bit trad, but I think of the teams. I've published quite a few papers. The end result of really cool research should be a publication, and I've published a few, and I've never done that as a sole author, which is, I think it's scary to publish by yourself. It's great to work as a team. So once I thought about adding up all the co-authors I've ever published with, and there'll be 1,000 or more. So I definitely think I've not met all of them. And that's because we work across countries. So some of my co-authors are in China or America, and I just haven't met them. But that makes me think, it's a great thing to be working in a team.
Absolutely. Yeah, science is definitely a team sport. So to put it in plain terms, these accolades are a really big deal. They recognise your distinction in research and your contribution to the advancement of science and technology and the depth of value and impact you have contributed to science. So when you think back and reflect, what personal values have driven this success?
Well, thanks, Hilary. You're making me feel all gooey.
Good.
I think patience is something that's forgotten. It often takes more than a year to get that paper out. So, if you're in a rush, you'll get really frustrated. You can't be in a rush when you're gathering the data either. You've got to see the end goal and visualize how good it's going to be. So you've got to be patient. In biology, you've got to wait for those results, the good results to come. You've got to aim high. So I don't like to just publish something because the least possible publication unit. I want to build a story and get it in a good journal. In a good journal, it will get cited more. It will have an impact, and that's always more fun than trying to go for little things that you've done. So be patient, let the story build. I think I know I'm a visual person, so I can imagine what the results would look like. Mm-hmm. That's not against the principle of science. You just got to see where your results will go and what they will look like in a figure, and then see whether the biology answers and gives you that result. I'm also, I guess, a bit sort of, I like quirky novelty, and so I'm always spotting things that are gaps in the knowledge. So I think one of my qualities, I hope, is that I can see novelty when it arises.
That's quite a skill.
Well, it sounds a bit like I've got pride in that, but it's just a skill to visualize where the gap is.
Yeah.
And science fills gaps, or we discover things that were wrong, and that's always harder to publish. If you find a result that's against a published previous result, goes the other way, or proves that a certain biological phenomenon actually doesn't occur, that's really hard to publish. And I've done it a couple times, but it's more painful. It takes a long time to overturn an existing result. So it's more interesting to fill gaps in knowledge and to go forward from what we've seen previously.
On the topic of patience, with the world, operating at a different, more rapid sort of speed, especially with digitalisation of everything, do you think that's being challenged as a skill set, the ability to be patient in science?
Yep. The funding cycles can be very challenging. If it's a 3-year grant, to be patient and wait for year 3 for your big paper can be terrifying because the grant's almost finished. But with artificial intelligence providing results from the computer that challenge the norm, you have to be faster. There are things in the laboratory that allow you to go faster. But faster all the time means possibly that errors creep in. So to tell a student, a good grad student or a colleague to slow down and get it right is a conversation that's difficult to have.
Absolutely. And so with values such as, patience and aiming high, that would require, I imagine, quite a bit of resilience.
Yes, especially you've got what you think is a good story, you get it out into the journal and it sits there with reviewers who are difficult for a number of months, and then the journal says, no, we don't like it, but if you could do some more work, we would reconsider it. That challenges your nerve to hang in there and not go to another journal and try something to get around the problem by going somewhere else. So it is scary. Another reason not to sleep at night.
No. That's not good to hear. I wonder how you've survived those 30 years of research with poor sleep. But speaking of the 30 years of research, take us back. What drew you to science originally?
Yeah, I've thought about that a little over the past few years, and I think even by the time I was a 10-year-old boy in Christchurch, I was already one of those kids who collects things in their bedroom, things that your mother doesn't want in your bedroom, like spiders and jars full of ethanol with some dead thing in it. Or, I was collecting rocks from various places around the South Island and having them in my bedroom. So I was already full of science as a boy, a collector. Bird watching. I was nuts about birds for a number of years. So by the time I was 15, 16, probably the bedroom was full of junk. And then I had the big benefit of going to the University of Canterbury as a young lad, just to be shown around the laboratories. So a university outreach into the school I was in. And, the university lecturers there just showed us through the labs. And I thought to myself, this is cool. I could put some of my stuff here. So I already had that. By the time I was graduating from school, it was in my head that I knew where I could fit in. So I think that's really important that the universities continue to outreach into schools and show the kids that there's a place for them. If they're quirky like that, scientists are a little quirky, which must have driven my mother nuts at the time. And that itch was satisfied by visiting the university and knowing that I could fit in there.
Yeah, that's great. What sort of laboratories were they that you visited on that?
Just general science ones, chemistry. I remember walking through the chemistry labs in University of Canterbury and thinking what strange smells there were.
Yeah.
And those old benches with bench tops that could survive acid spills and all that sort of stuff. Also, I was already doing it at high school well in the science subjects. General science. The only science I wasn't very good at is physics, which is strange because my maths was doing quite well. But physics was always a hurdle for me. So I followed my nose into biology.
I feel I'm in good company. Physics was also my weak point.
Yeah, we're not being rude about physicists. They're amazing. No. But I think my visual brain just didn't suit the way the data was thrown at my head, basically. And I think biology, chemistry, even statistics, I can visualize things okay. But with physics, it was a challenge.
So you chose to study biochemistry and molecular biology. So how did you end up with those subjects as a priority?
Straight out of high school into Canterbury, took a broad science set of courses. So everything from stats to chemistry to... I didn't do physics. I knew that would end badly. But I did every other topic, including zoology. There was a thought that I could go on to medicine, but in the end, I just knew my passion was in things that, like plants, that were biological and they didn't. I found dissection of animals quite tricky, so I found a passion more in the plant biology science. I love genetics, and genetics in plants, as shown by Mendel, is a lot easier, and you can do the numbers a lot better, and the maths comes out better. So I found a happy place there. So I started broad, and then I narrowed things down as I followed good results. So often the things that you did at uni that you love the most, you'll get the best grade in, that then goes on to the next year and you keep getting better and better grades. I would recommend follow your passion, the things that are delivering well, and you'll end up in a more focused area that you didn't know you'd end up at the very beginning.
Nice. And so following that focus, is that what led you to your PhD?
Yep. At the end of an honours degree at Canterbury, I'd done really well in plant biology, genetics, biochemistry, and there was the opportunity to look for a scholarship funding to go overseas, which I then did.
Okay, so tell us a bit about your PhD and your experiences overseas.
It started with applying to a lot of different funding sources, and that was scary because the funding cycle at that stage was right in the middle of when we were meant to be studying for our exams. So I think I did a month of really hard work, both doing the exams and applying for lots of funds. And I was very fortunate to get a scholarship to go to Cambridge. So off I went to England, not knowing very much about England at all. I tried to do some SWOT about Cambridge University by going to the library, and all I could find were detective novels about Cambridge where half the population was murdered. But so I arrived in little old sleepy Cambridge, England, not knowing a lot, and fell in love with the place, loved it. So 3 years PhD scholarship, had to finish after 3 years because the money just runs out. So I had to round up my research as quickly as I could, write up a big thesis, get examined, and then get on to the next thing.
And what was the topic of your PhD?
I worked on auxin, a major signalling molecule in plants. So auxin controls growth, apical dominance, the dominance of growth in the top of the plant. So we worked on how that compound moves within the plant.
Okay. And then where did you go after that?
I moved on to a postdoc in Edinburgh, where I looked at the responses of the guard cell, which is a cell in the leaf of the plant which opens and closes in response to drought. So I was basically working on drought tolerance in plants, but the key hormone that controls that movement of the stomatal guard cell is Scigest acid, so another plant hormone. So I worked on the signaling of that hormone for a 3-year postdoc.
Okay. How many postdocs did you do until you got yourself a permanent position?
Another postdoc was done in Israel in the Weizmann Institute on the signaling response to pathogens. So I did a bit of plant pathology at that stage. So 3 years in Israel, in the heat of Israel, which was very interesting. There were 2 postdoc journeys, if you like. And then in the final year of that postdoc, I decided to start looking for a permanent position.
Is that what led you back here to New Zealand?
Yep.
Right.
So that's the issue. You focus down to a very specific area of plant biology in this case. So the number of positions that will pop up will be counted on one of your hands, even if you look all around the world. And so I was doing that, and there were options in Australia. There was one decent job I went for back in Canterbury, and there was a position here in Auckland. So I had my CV. Always get your CV well prepared. So I had my CV with about 3 groups. They were mulling it over, I guess, and I managed to land one of the 3. So that got me back to Auckland, which was a pleasure. I wanted to be back in New Zealand. I missed the place, been away for almost a dozen years, and it was time to. I was having some family, some children. Well, I wasn't. My wife was. So it was time to let my roots grow, and I wanted to be back in the country. So I was lucky. So
you ended up here at what was Hort Research.
That's true.
In Auckland, right?
Yeah, 1998, I think. It's a blur because, as I said, I was having children, so I wasn't sleeping at all. And it was a busy old time. So it was great to be back. I got myself established. Kids started to go to schools. And the intention perhaps to continue with the science career, which involves moving if you want, that intention started to get sort of more based in reality once kids were in school, because you can't rip up your family and move every 3 years when you've got wee ones relying on you.
Yes, that's right, a good bit of stability. And so what was the job that you first got at Hort Research?
As a scientist. So early on I was working with the response of plants and fruit in particular, because Hort Research was focused on horticulture. But my interest was in again, in abiotic stress response, the tolerance of a plant to stress that is environmental, like temperature. So the post-harvest group here in Auckland was working on cold response, on what happens to plant cells when they go in the cold, and that was one thing that was very interesting to me. So I was helping out in that area.
So a lot of work in your early career on abiotic stresses then.
Yes.
So quite useful sort of grounding for the times we have now.
Absolutely.
Yeah.
So with the climate crisis, the plant response and how it copes to extremes in temperature, we're talking generally things are heating up, but we'll get weather events where it gets a lot colder, a lot quicker, extreme weather events that are short and acute. And the plants that we grow need to understand how they respond to that so that we can make our horticulture and agriculture sector more resilient.
Indeed. There was also a lot of application for new jobs, applying for a scholarship in the middle of exams at university and then moving around to different postdoctoral opportunities around the globe. So that transient nature, is it something that most scientists should expect to have in their career?
You can think of it as a positive and a negative. The ability in science to be international, to go to different places and talk the same language, even if that place their native tongue is not English. Fortunately for me, often the science that we do is in English. So you can be in different places and do some amazing stuff. In the end, you do want to get some stability. So the postdoctoral system where you do short contracts in different places is exciting, it's cool. Then eventually you need permanency. Otherwise, how can you buy a house and how can you live and have a family? Doesn't stop you travelling. Once I started to settle in Auckland, we've done a lot of travelling around, and so that's cool. It is really neat to see different countries and do that under the banner of the science we do. The benefit is that when I travel, I meet people, I bring the techniques back to New Zealand, we benchmark what we do better science because we've got links to everybody.
Yes, some strong benefits there. You are also professor at the University of Auckland. So how did that arise and how do you balance the needs of the two different roles at University of Auckland and plant and food research?
I think the very early 2000s, I got approached by the university to do teaching because there was a gap to fill. They had a plant physiology course running and almost no one to teach it. So I jumped in with very little notice and covered, and that covering their teaching has extended for another 22 years. So obviously it worked out. I've achieved a teaching record now, hopefully explaining to the undergrads how exciting the field is, and I think I've done all right teaching. How do I balance the competing jobs? Well, one, you don't get bored so easily if you're doing a bit of both. So if I was just a researcher at Plant and Food, then I probably would have got bored quicker. If I was just a lecturer and not doing any research, I would have got bored quite quickly too. So having a diversity of tasks in your year or in your day-to-day life means that you don't run out of energy so quickly, as long as you stay sane. So you can't work double the amount of hours of anybody else. So you need to learn to say no.
Yes.
So if tasks come up in the different institutes which don't suit me, and if I was a sole worker in a single institute, I'd probably have to say yes to the tasks. I can quite, in a valid way, say no to a certain task. So tasks include things like working on committees that control, purchase of equipment, or these are science tasks that have to be done by scientists that aren't research and that aren't teaching. And I tend to be able to say no to certain things. Though my team members say I say yes too often, you've got to have the ability and the strength to say no to certain things. That's
Absolutely.
how I stay sane.
Yeah, for sure. And there's a time and a place as well, because it's also good to take those tasks on to get out there more, to get involved,
Yes.
with the wider area.
Exactly. And I've done various roles, committee roles and leadership roles, which I've stepped back from, because there's no point doing it for the rest of your life. I've had positions, science group leadership, which I've run for 5 years and then stepped back from because I want to do other things.
Yeah.
So if you're doing something new, you've got to look at your inbox and say, well, what am I going to retire from if I'm going to do something new? What task am I going to stop if I'm going to do a new one?
Mm-hmm. Yes. So let's talk about your research. What have been the most enjoyable science questions you've explored, and why were they so enjoyable?
I think the colour response work that I've done, we're looking at the apple response to cold, and one of the things apple does when it gets cold is it goes red, which is pretty weird. But the apple fruit, late towards harvest, if there are cold nights and sunny days, then the colour of the skin goes bright red. If it's too warm and cloudy, then that colour response is less. So we started, because I was interested in temperature and abiotic stress, we were just using the colour response as some sort of visual assay of that stress, something that we could just see. And it actually became more interesting than I expected. Just what makes things go red became a sort of obsession for a dozen years. And everybody loved the results. We discovered genes that control the colour response in apples and kiwi fruit and cherries and roses and all sorts of things because I was studying this fundamental on-off switch for colour. What was the benefit is that we were working on a thing that the consumer loves, that the grower loves, and that biologically a molecular biologist just loves something that goes on and off, and that you don't have to measure with a gel or a PCR machine. You just can see that the response happened. So we had a lot of fun with that. Fun, you should enjoy your research. You should also be able to explain what the impact of that research is. And we could with the colour story. Cultivars that are really red kiwi fruit in our supermarkets, these are high-value cultivars that they add novelty to the purchaser. They add health compounds. So anthocyanins, which are responsible for red, are healthy. They're good for you. So there's all these ticks in the box of benefit which we got from that research. So I benefited because I published well. They were great papers that we were getting out on the colour response in lots of different plants. I benefited because I had new mates that were growers, and I had new mates in the industry, and we were doing work that was visually exciting, which is what I like doing. So that was neat. Now we're working on the climate crisis, and that's a hard one. It's hard to be positive about the issue. Yes.
Yeah.
So I want to remain positive. It's not frivolous either. It's not something which is just novelty and exciting for the consumer. It's serious stuff.
Yeah.
So I think we can help big time. Biology can certainly help the issue, and we have to remain positive. So it's more serious than colour, but the climate crisis is serious on a different level.
Yes, indeed. So what are you working on at the moment?
Yeah, we've got a nice Endeavour funded project on flowering, and it's the effect of high temperatures on flowering. In particular, most of us consumers wouldn't realise that in midwinter, our temperate crops, our kiwi fruit, our apples, our cherries, the buds are asleep, waiting to break out in spring. But while they sit there dormant, they're actually counting the number of cold nights there are. And that counting, the little clock inside the bud, if it doesn't get enough of those cold nights, then in spring it will burst as a group of leaves and no flower. And so if your crop is relying on those flowers to make fruit, then you're going to get less and less fruit. So that's an aching problem of climate change, is that our winters, as they get warmer, will reduce our horticultural productivity because of this need for cold nights in the middle of winter. This is a big problem.
Yes, absolutely. So what's the solution?
The solution is the genetics. In peaches, for example, with 100 years of breeding, there are peaches that will grow and flower and fruit in hot places, and peaches that will grow and flower and fruit in central Otago. So they've done well breeding new versions of peach that can produce peaches everywhere. with kiwi fruit, cherries, apples, we need to start figuring out what those genes are and get breeding new cultivars that will be able to cope with warmer temperatures, unless we want to move production somewhere else. And, that's a problem. We don't want our kiwi fruit growers to stop growing kiwi fruit and have to grow something else because it's too hot. We need to supply them with new cultivars which can cope with those warmer winters.
So in terms of breeding things fast enough for the rapid change in the climate crisis, what tools are in the toolbox to help with this? Breeding can take quite a long time. So what do we do?
That's where we get to the edgy bit.
Yeah.
So the discussion on changing genetics so we can have a plant that is able to grow in a warmer climate, which is happening, means that we can do those changes in genetics by crossing, normal natural breeding. That can take sort of 7 years per generation, per cross. And if we have to do 4 or 5 crosses, then we're in 2050 already. If we're looking forward, the temperature change is too quick for our breeding programs.
Mm-hmm.
So I think we need to use new, faster techniques. And that doesn't involve transgenics. That doesn't involve putting a new gene into a fruit. It involves just speeding up the breeding program using some new tools. And one of those tools is fast flowering breeding programs, so individual plants that flower all the time, so you can do crosses really quickly. Another approach is CRISPR, which is to make edits in a plant, little changes, variants in key genes using this system that doesn't add DNA, it just makes new genetics, if you like, that can be used for the new environment. I want to reiterate, CRISPR doesn't add DNA to the plant. Mm-hmm. It just changes key genes that we know something about that will give us variation that we can test to see whether it can cope with hotter winter nights or even hotter summer days. We need to understand our crops, make sure they're growing better in the changing environment. That means they need different genetics.
Right. And so there are various tools we have now that can help introduce new genetics, so we're not sifting through trying to find it in nature. Yeah.
And they're talking about this topic triggers some people. A scientist has said, I'm going to change genetics, and that upsets a portion of the population. The thing is, though, genetics change all the time anyway.
Yeah.
If we look at, breeding of dogs, there have been lots of DNA changes caused by taking one Pomeranian and crossing it with another Pomeranian.
Yeah.
So we're okay with changing genetics in a natural way that we've done over the centuries. All I'm saying is we can do the same a little bit quicker because of the problem is coming at us fast.
Yes, that's right. So gene technologies, have they been a key enabler of your research then in the lab?
Oh yeah, yep. It's an amazing tool to prove how. I'm a physiologist, so I don't really look at DNA sequence very much. I like to say, well, I think this major gene in this plant is involved in whether it's red or not red, and then to go in there and make a variant of that gene, and then wait for the plant to produce some fruit, and they're not red anymore. That is very good proof that you were right. The hypothesis was right. That gene is involved in being red or not red. Mm-hmm. So it's a great tool in the laboratory. Then the product of that event would be quite interesting as a new cultivar. That's the issue. However, the public are not happy with the results coming out of the laboratory and being planted in the ground. Not happy at all.
Yes, it's a big topic, and with new potential legislation changes under a new government, it's probably a topic that New Zealanders at large will have to start discussing, I suppose.
We have been in conversation for a few decades now, and it goes in circles. So we're in a circle again at the moment.
Yeah, absolutely. There's a saying, the greatest threat to our planet is the belief that someone else will save it. And many people look to scientists to have the answers and provide solutions. In your view, do you think the research, science and innovation sector is doing enough to solve climate change?
I would have said straight away no, because I would like more effort and funding into the process. But I don't want to be too negative. I think around the world, scientists have rolled up their sleeves and they're working very hard on this. So if as a global community, I think that we're doing okay. I'm trying to be positive, that we need to understand that there's no magic one bullet. There's a number of things that are happening, and we should try all of them within safety bounds.
Yeah.
With the climate crisis, I think a third of the solution is changing our energy use, so move away from fossil fuels to renewables. A third has got to be some big engineering projects that I'm not an engineer, so I don't fully understand, so not my area. But I'm a true believer that a third of the solution is in plants, which is amazing because I work on plant biology, so that's exciting. And I'd say a third because I don't want a third of the funding. Plants are amazing. They fix carbon dioxide. So fixing carbon dioxide is the major issue. We've got to get it out of the atmosphere and back into plants. The other thing is plants will do that. A more plant-based economy will fix more carbon. Plants also produce food. So as we fix carbon, we produce more food, and we need that too. So plants are, and they're also the most efficient capturers of energy. The photosynthetic system is the most efficient system ever developed in capturing light and converting it into energy. So they're amazing little things, or big things. Some of them get quite large. So plant biology is a big part of the solution. We around the world are working hard enough to try to get those solutions forward. Are we doing enough in New Zealand? The answer is, I think we need to refocus some of our research on this issue. And as we produce some tools out of that research, we've got to use them.
Mm-hmm. Yes.
So I started off with a no, and then I think to be positive, the science community is doing the hard work now.
Good. That's reassuring to hear. A long time ago, in a lecture theatre not far away from here, you recommended a good read to me and the class I was in. It's called Tomorrow's Table, Organic Farming, Genetics and the Future of Food. And to quote from this book argues that a judicious blend of two important strands of agriculture, genetic engineering and organic farming, is key to helping feed the world's growing population in an ecologically balanced manner. What does the future of sustainable food production look like to you?
It's a very funny book, that one, and I still lecture about it. Pamela Ronald's one of the authors. She's a hero of mine, a molecular biologist, and her husband is the co-author, and he's a lecturer in organics in California. So the two of them fight with each other on the page about what's sustainable and what's not. So it's a very good book. And in the end, they're still married. So the fight must have worked out quite nicely. The answer is that molecular biology and moving genetics is organic. So I don't understand why the organic sector has fallen out with the thought that changing DNA is a bad thing. It's very odd that it's become two bubbles that are mutually exclusive. They actually overlap very well. If you get good genetics from whatever means, it's an organic solution. It can reduce pesticide use, it can reduce water use, it can save us from the climate crisis. So I think the two ideas, the two concepts need to talk to each other well. And this book illustrates how that conversation can happen. The future of a sustainable food production is produce on the existing amount of land that we have, at least maintain the yields on that land in a changing climate, and if you could, increase the yield. So that sounds like it's not sustainable, but with new techniques, it's got to be. We've got more and more people on the planet. That increase will gently start to decline. It's an interesting way of saying it. But the increase, the increase won't be as large over the next few decades. We can't reduce the world's population without doing something horrible, okay? So we're not going to do that. We've got to work at the target of not having more and more people, but the people that are on the earth at the moment need to be well fed. So we can't reduce agricultural production. We've got to maintain it, but have less impact. So new tools need to be used. To be truly sustainable, we need a more plant-based agricultural and horticultural system. We have to reduce the amount of meat and dairy we eat globally. And that's a hard one for New Zealand to get their heads around because we're making good money out of meat and dairy. But in the end, for the benefit of the planet, we need to move towards a more plant-based food system. And I'm a bit of a hypocrite there in the fact that I eat meat, but my personal goal is to eat less and less meat and just have meat for special occasions.
Yeah, I agree completely. And it doesn't have to be total elimination of something. It's just more moderate consumption.
Yep. And New Zealand can produce high-value meat and dairy, but we've got to do less of it, and we've got to make sure that our farmers are still very successful by giving them options, plant options, where they can make good value. And that is the challenge for our science sector, is to provide those options to our economy so that our farmers and horticulturalists can grow new crops that are going to give them good money, and therefore they don't need to convert their land to dairy.
A lot of discussion needs to be had. And you mentioned about people's perceptions of gene technologies earlier, sometimes good, sometimes bad. So in your view, how do we move forward with good judgment and sense to make the most of new gene technologies to reduce the human impact on the planet?
I've thought about it a lot. I've tried the education approach. I've given lots of lectures on the topic, and it's okay. I tend to talk to people that already wanted to listen to me. No, I don't tend to. That's what happens. The people that come to my talks usually are the ones that want to listen to the story. Those that are really anti-gene technologies are not going to listen. So the remit I now think scientists should have is to keep producing really good examples. To produce a plant or a concept that is just a game changer, that the public will say, well, I really want to try that. So that's my approach at the moment, is to keep talking about what I'm doing openly. We've got to be very open, but also produce a cultivar or a plant which is the product of some of these new technologies and say, this is way better for the following reason.
Yeah.
This plant is way better because it will flower in a hotter climate, and we're getting a hotter climate. Or this fruit is way more interesting because it's got a health compound that is really good for you, and it's going to make us good money. So we just keep rolling out really good examples. That's my goal, and that will eventually give us that conversation more fuel in a positive way to move forward.
You've mentioned your role in lecturing before, and I've been a beneficiary of that. I was one of your students and I enjoyed my lectures with you. So you've been pivotal in developing the next generation of scientists. And to feature on the Clarivate Highly-Cited Researchers list, I imagine one needs to be both a good team player and team leader. So what makes a good team leader, in your opinion?
Sometimes scientists aren't the best people. They're not as personable as the general population. We can be very introvert, very focused, and not good at explaining. So I think I've run teams. I've got the skill to talk things through, I hope, I try not to appear too arrogant here, because that's tricky for me, is to think that I'm any different from anybody else. However, when you're in charge of a team, there are responsibilities to explain stuff and to make some decisions and to get them across the line. So you've got to, I wrote down trust is a big thing. So I've got to trust the people in the team that they'll get the result done, and they've got to trust me that I'll help them and that I'll explain to them why it's so really important. So there's got to be a two-way support of each other, and that comes from trusting each other in the tasks. So I think that's really important. I think I'm humorous, so you've got to keep laughing. So, yeah, the team has got to have a giggle every now and again, let loose, celebrate success. If the paper comes out, then have a good old party or something. Have a break and celebrate that. Finally, I'd say keep up the enthusiasm. If there's a big issue, I always just get in there and help. So I'm often alongside the postdoc or PhD student, trying to help. So that's the way you run a good team, I would say. If there's a lot of work to do, just get in there and do it too.
And get your hands dirty, which I literally saw you doing the other day,
Yes,
getting your hands dirty with some soil.
I'm less in the laboratory these days. In fact, people laugh at me when I go in there, but I am in the greenhouse all the time with the plants because it's a great release of tension being down there and just potting around. So that's my practical time at the moment, is down there in the greenhouse.
Nice, which provides a lot of joy, I'm sure. So looking back on your incredibly successful career.
Stop that.
What do you think are the secrets to a successful career in science?
Yeah, I don't want to reveal this. No, of course.
Give it to yourself.
Yeah, it's not a secret if I'm going to tell you. But I think the art is one of the things I learned early on is what my own rhythm is, in that I know I'm a morning person. So I start quite early, and with coffee, I do really well in the mornings. And that means, what does that mean? It means in the mornings I should protect the time I have in the morning to do the clever stuff. And if somebody out there is dropping a lot of appointments in the morning for me, I know I'm not going to perform as well. If it's a meeting that I've got to be sharp in, like today's chat, then that's fine. My energy is high. In the afternoon, my energy declines. So I find myself doing tasks that are more routine, that are less requiring being sharp. So be protective of your time when you know you're at your best. Then that equates, I think on a day-to-day basis, I spend about a third of my time in the greenhouse doing the science. I spend about a third of my time in front of the computer writing about it, editing. A lot of work these days happens on the computer. So I'll be on there doing work, but it's in front of a screen. So getting away from that requires a break. Do that. So that's a secret. Have plenty of breaks. About a third of my time spent with people. So that divides up the day. So that's a secret, is to keep yourself going by making your day very variable. The final secret is have a weekend.
Yes.
Okay? So a lot of scientists work way too hard and they lose their personal time. In the end, that will cause you to unravel. You will go backwards if you think you can work 7 days a week. So the real art is to have a good weekend, and in that weekend you're still thinking about your work, but you're not at work. So the brain is still chucking away, problem solving over the weekend. So when you get to work on a Monday morning, you're fixing things. So that's the art of a long career, is to stay sane and make sure you protect your personal time.
I'm hearing a good dose of self-awareness and boundaries and rest.
Hard-fought self-awareness, because I have spent times in my career where I've worked 7 days a week constantly, and I just end up not sleeping, performing a lot less than I should be, but I thought I was doing well, and it's not the answer.
Yeah, you have to learn the hard lessons sometimes, sometimes multiple times.
Yes.
Well, it's been a real pleasure speaking with you today, Andy. Thank you for taking the time to share your story and your research. We look forward to hearing more about your exciting and important science in future episodes.
Thank you so much, Hilary.
And thank you out there for listening. If you'd like to know more about the topics covered on Scigest, check out the episode pages on the Plant and Food Research website. Please also subscribe to or follow the Scigest channel and rate and review each episode. Why not give it 5 stars? Thank you, and mā te wā.